Hello my dear students, this is Dr. 𝗲𝗵𝗺𝗺, your biochemistry faculty. Well, in this video we are going to discuss about RNA. What are the different types of RNA? What is the nature of RNA? Okay, so this video is about RNA. Now, this RNA stands for ribonucleic acid first of all. Right, ribonucleic acid. That means it has sugar as ribose, right? In contrary to the sugar which is present in DNA, which is deoxyribose. So this ribose is the sugar which is present, and generally it is single stranded. But yes, exceptions are there, stranded, but exceptions are there in other, exceptions in viruses. Then this RNA has uracil instead of thymine. This is also very, very important. So it also forms base plus sugar plus phosphate, which forms a nucleotide. Similarly, another is base plus sugar, where no phosphate is there. It is known as nucleoside. Right. Now, coming over to this, there are so many different types of RNAs, right? But major we focus on messenger RNA, we focus on transfer RNA, and we focus on rRNA. Right, these are the two main. mRNA stands for messenger, T stands for transfer, and R stands for ribosomal RNA. So these are the main principal types, right? So other types of RNA also I will just show it to you. You can see that messenger RNA is there, right? And ncRNA is also there. ncRNA is a non-coding RNA. mRNA is a coding RNA. Okay. So I can write here because it is basically bearing the codons, or it is helping in the transfer of the messages. Or non-coding RNA is ncRNA. Then this ncRNA is further of different types. If you see the types clearly, housekeeping ncRNA is there and regulatory ncRNA is there. If we talk about housekeeping, जैसे अभी I told you ना कि 3 RNAs are very, very important. So other housekeeping non-coding RNAs are transfer RNA, ribosomal RNA, and regulatory are, you can say, this RNA long non-coding, or it is small non-coding RNA, right? And then this small non-coding RNA consists of microRNA, you can say small nucleolar RNA, silencing RNAs, small nuclear RNA which are also known as SNRPs, and PIW interacting RNA. So because these are the ones which are sometimes used as gene expression, okay, or gene regulation. If we talk about silencing RNA, they have their own function. If we talk about small nuclear RNA, they have their own functions. So this is the different categories of RNA. But the main ones which we can say housekeeping are transfer RNA and rRNA and messenger RNA, which is the coding one. So coming over to this, so if we talk about the structures of this mRNA is having a linear structure. Always remember, this mRNA has a linear structure. Which means it has this type of structure and it bears the codons. So these are what? These are the codons, and that is why it comes under coding RNA. Codons are there, or you can say these are the coding sequences or the exons. Then we come over to transfer RNA. Now this transfer RNA has a clover leaf-like structure. clover leaf-like structure. And this structure is, it is having this type of structure. 5 prime, 3 prime, like this. Like this, a labile arm is there, that is why clover leaf, and this is there. Now the most important point which we have to remember here is that this is the 5 prime end and this is the 3 prime end where CCA is there. Right? CCA is, or the 3 prime position is, which carries the activated amino acid. Activated amino acid. Right? This is bearing the anticodon. This is pseudouridine and this is DHC. So this is how, this is the structure of transfer RNA, which looks like a leaf. Labile arm is there, pseudouridine site is there, you can say there is one enzyme recognition site here, and the most important point which you have to remember here is that 3 prime end may the activated amino acid is there. Very important, right? And then the third structure is of ribosomal RNA. Now, ribosomal RNA is the one which is present in abundance. Maximum concentration is of rRNA. Now, if we talk about rRNA, ribosomal, so we say that prokaryotes and eukaryotes. If we talk about prokaryotes, they have 70S and eukaryotes have 80S. So 70S is further 50S and 30S, 60S and 40S. And what is S? S, it is simply the Svedberg unit or sedimentation coefficient. That means the ribosomes have 2 subunits, a smaller subunit like this and a larger subunit like this. Okay, so this is the smaller subunit and this is the larger subunit. So the larger subunit is having 2 different sites. One is the P site, other is the A site. Now what is the difference between the P site and the A site? A site is the acceptor site. Right, for amino acids. Right, because it is going to accept, and P site is the peptidyl transferase site. Peptidyl transferase site, which is required for the formation of the peptide bond, because this is, we are just making the infrastructure ready to undergo translation, because first replication is there. Replication is altogether a different process where RNA is forming RNA. Transcription is there where this DNA is forming RNA. Then that information which is hidden in the form of a genetic code, that has to be translated, and it is translated by a process known as translation. Okay, so for that the entire infrastructure has to be ready. So we can say the principally important RNAs are mRNA, tRNA and rRNA, which are the housekeeping RNAs also. Among this, only mRNA is the coding RNA. Rest of them are non-coding. And rest, this is the classification which we have already discussed. And always remember, whenever we talk about the other RNAs, they are helping in gene regulation and expression. Regulation and expression, very important, right? So this is all about RNA. Then after this, then in the next video we are going to study about translation, which is altogether a different process, where how the information will be translated and how this messenger RNA, you can say tRNA and ribosomal RNA are going to give their contribution. So that's it. Thank you so much.